I received my first AA&MDS grant when I was a fellow, still in training, when I was in the laboratory of Lucio Luzzatto at Sloan-Kettering. And that was really the project that first got me interested in aplastic anemia and paroxysmal nocturnal hemoglobinuria and the relationship between those two diseases. And so I was a recipient of the New Researcher Award, and I believe 1998 was the first year, and then I think I had a second year of funding in 1999. And I remember attending my first conference, I believe the year before, in 1997, when I was first getting into the field. At this stage in my career, I'm also the recipient of a current grant, and this has now enabled me to do some work where we're asking other questions about PNH, particularly whether there are other mutations other than PIG-A that are mutated in a subset of patients with PNH, and also to try to get a sense of whether we can predict better who will get blood clots. And you know, we do flow cytometry to diagnose PNH routinely, and we do it on the cells that we can do this very easily, red cells and granulocytes. And we are now in the laboratory developing a simplified approach to looking at platelets also, because there are PNH platelets that we don't really think about so much because we don't get back a test result saying what are the percent PNH platelets. And we're now, others have tried to do flow cytometry on platelets, and we're modifying that just a little bit to try to get a cleaner separation between the two populations. And we think that this will help us get a sense of the PNH platelet clone. And it's our hypothesis, we will see whether this is a better predictor of blood clots, which is the major complication in patients who have PNH.
So then you can manage.
I think that early involvement with the foundation was really instrumental on getting me involved in a career in research and also clinical care of patients with bone marrow failure diseases, and really in a number of different ways. In terms of the research that I was doing at that time, it supported work to see whether normal people have small PNH clones. Now, this is actually something that we talk about now. Oh yeah, normal people have small PNH clones, but we didn't know that at the time. And what this involved was looking at blood samples from normal people and seeing if we could see the same types of cells that you pick up by flow cytometry in a patient with PNH or aplastic anemia. When you get an abnormal result, we just here look at millions of cells instead of, say, 1,000 cells. In someone with PNH or aplastic anemia, you look at 1,000 cells, and you might see, in someone with PNH, you might see 50%, okay? And you would pick that up right away. In someone with aplastic anemia, you might see 1%. So with 1,000 cells, that would be enough. In a normal individual, you would have to look at 1 million cells, and yet you pick up a couple if you look at 1 million. So we just scaled everything up, and we found those mutations by flow cytometry. We found the mutants by flow cytometry, and then we collected those cells and did genetic analysis, the same type of genetic analysis that was being done in the laboratory on the patients with PNH. We did it in a little bit more detailed manner, and we found the same types of mutations that cause PNH. In one case, the same mutation that's known in another lab to have caused the PNH clone. And this was very important because we did not know at the time whether the PIG-A gene mutation was enough to cause the disease PNH. But by finding these types of mutations in really all normal individuals, it argued strongly against the idea that the PIGA mutation causes PNH in and of itself. And this was important because there's another set of diseases, the leukemias, where you have a different kind of mutation, where you have a translocation, where one chromosome attaches onto another, and that probably is enough to cause that type of leukemia. And here, this was strong evidence that PNH was behaving in a very different type of way, that it really does not belong in that category of diseases. And it lends support to the idea that it's the immune attack against the marrow in aplastic anemia that causes these PNH clones to come up in aplastic anemia and in patients who have PNH, because there's this relationship between these two diseases. And this finding really helped support that idea. One of the things that's been very gratifying is that sometimes patients will ask me about this. Do normal people have PNH clones? And I would be able to tell them, well, yes, thanks to this work that was supported by the foundation early on, we can now say, yes, normal people do have this in minuscule quantities. There's been that work that was supported by the foundation also helped research that I started in on in terms of expanding this, because the idea that normal people get mutations in all can get a mutation in any gene is really important for a different type of disease, namely cancer. And it has given us some insight into whether cancer cells have more mutations, and it has opened up some avenues of research in that way. And it also has been important now in terms of testing drugs to see if they cause mutations. And this is work that other people have done, taking on this type of project that I started. They've been looking in animals, injecting mutagens with the idea that this can help the pharmaceutical industry determine whether a drug is safe by looking to see whether it causes these mutations. So I think this is an example where the study of a rare disease actually really helps us to understand very common diseases and gives us a tool that is important in a much more general way. I think that one of the hopes, as I mentioned, was that we may be able to predict better who gets blood clots. Another hope is that we may be able to separate out small subsets of patients who have an overlap syndrome between PNH and myeloproliferative diseases. So I should maybe step back a little bit. There are these overlap syndromes. We sort of draw these Venn diagrams with these circles overlapping each other, right? There's PNH and aplastic anemia and MDS, right? So the overlap between PNH and aplastic anemia, there's no question that maybe 40% of patients with aplastic anemia will have a PNH clone. Maybe a similar percentage of patients with PNH will have clinically important cytopenias that we say low blood counts. The overlap between PNH and myelodysplasia is something that I actually worked on also in those early years when I was a fellow. And we basically showed that patients who have PNH very, very commonly have changes in the bone marrow that are reminiscent of myelodysplasia, but they never develop myelodysplasia. And so we've sort of there still is an overlap because some patients with PNH will develop something that's very, very clearly an advanced form of myelodysplasia and will require treatment for myelodysplasia, but those are really a small subset. Now we're asking the question about this myeloproliferative disease, which is a disease which is the opposite of aplastic anemia, really. You have too many cells. You're making too many white cells, too many platelets, in some cases too many red cells, too much of a good thing, right? And we found a couple of patients who have a mutation in a gene called JAK2, which is associated with this myeloproliferative syndrome, too much of a good thing, right? And sure enough, these 3 patients had features of myeloproliferative disease on top of, no doubt, PNH. And what was interesting is that if you go back to the literature from the 1970s, there were a couple of patients who were reported who had that overlap syndrome. Nowadays, we can confirm that genetically in ways that they couldn't back then. And so there really is this small subset that have that overlap syndrome. And we are now looking at these genes that are associated with myeloproliferative syndrome, and it's possible, well, we do think that there is this subset that will behave a little bit like a myeloproliferative disease. I have spoken at some of the patient conferences that the foundation puts on, and that has been actually a very gratifying experience for me to be able to stand up in front of patients and tell, and these are patients who may have never met anybody who have aplastic anemia or PNH. This is not like diabetes where your next door neighbor has the same thing, right? This is something where it's almost unheard of that you would have met somebody who has this. So these meetings get people together, and they get presented with information that you might hear at a medical conference such as this, but put in a way that patients are going to be able to understand. And that has been very gratifying. And then in recent years, I've started to put on webinars for the foundation. And that's basically the same thing, only it's a virtual online conference lasting really 1 hour. I give really a 45-minute talk using slides and which people can see on their computers, and then fielding questions afterwards. And if I'm not mistaken, I think that these webinars are still on the web page, so that they're recorded and they stay on, so that people can go back to them. Even if they weren't participating in the webinar, they can go and look at it later. My involvement in the foundation also was in the early years when I was getting involved clinically in taking care of these patients. I would get calls from patients who were in a different part of the country asking for advice about the management of their care with either PNH or aplastic anemia. Maybe they were not near a center that had expertise in this, and the foundation knew about me because I was a grant recipient, and I would be able to help some of these people long distance to, how do we say, get plugged in to a place that could help them. I think that for fellows starting out, going into hematology and oncology, that there are a lot of things tugging fellows more towards oncology, and hematology, which used to be more a bigger piece of that combination. I think that it proportionally is becoming less, simply because oncology has gotten to be such an exciting and tremendous part of that combination. So I think that the foundation, in terms of supporting young fellows in something that's still in the hematology part, I think that that's very important to maintain that balance and to ensure that there will still be fellows who are interested in going into the hematology side, even though oncology is now clearly very exciting and very promising, and now everything is changing for the better in oncology. We still need people to go into what used to be the major part of the field, the hematology side. I would give them encouragement to say that they should definitely seek out all possible sources of funding. The government sources of funding, for sure, everyone thinks of that, but also to think of foundations such as the Aplastic Anemia Foundation. If they're going into solid tumor or malignant hematology, there are other foundations, and that they should definitely consider all possible sources of funding, not to be discouraged, because this is a difficult funding environment. To reapply if you don't get something the first time, to revise and reapply and to stick with it. It is said, for good reason, that it's hard to do research and take care of patients at the same time. And people going into research, particularly people who have MDs who are going into research, who still are going to have a clinical involvement, you're going to hear that time and time again. And the reason you're going to hear that is because it's really true. It is a challenge. And yet there's great value to it, because there is this talk about these overlap diagrams, right? You've got the clinical and you've got the research, and in the intersection between the two, you have ideas that get generated that do not get generated when the two are completely separate. And there's that great role for what we call translational research. And as hard as it is to be on that edge of two different, very different fields, if you think about it, two different, very different types of careers, endeavors, as hard as it is to be on that edge, try to stay there and try to get into that edge and see if you can come up with ideas that the clinicians can't come, the pure clinicians and the pure researchers couldn't come up with on their own.
Great.
That this is a rare disease, and it is people who dedicate themselves to raising money for a foundation like this that really help keep this on the map. And they have a tremendous role to play, and more so in a way proportionally than for another disease. For example, you know, in common diseases like asthma and diabetes, there's huge pharmaceutical interest in those diseases, and proportionally a lot of resources that are available. For here, I'm not going to say that this is it, but this is proportionally much more important to the field than it would be, for example, than a similar foundation might be for these very common diseases that are, you know, household names. So I think that it is crucial, and of course it is exceedingly appreciated. And I think I could speak for everyone who has been a recipient whose work has benefited from the funding of the foundation as a way of thanks.